Hello, everybody. We're going to look at glycogen metabolism today. We're looking at the synthesis and degradation. And so we've talked about maintaining glycemia, how it's really important that you want to be euglycemic. You don't want to be hyperglycemic. That could be diabetes. You don't want to be hypoglycemic because that can cause a lot of problems too. And we mentioned there's three sources of blood sugar. Today, we're going to look at glycogenolysis and glycogen synthesis. So what you do is you go to bed at night, maybe midnight here, and what you do is you have falling levels of blood sugar, and the glycogenolysis here comes down. You have gluconeogenesis, which you've talked about, and you can eat dietary sources of glucose. So we'll take a look at glycogen. And we know that glycogen regulation will be controlled by the relative levels of hormones, such as insulin and glucagon. You should always think about those as opposites of each other. So in the fasted state, you have a low insulin and high glucagon and high epinephrine, as well as cortisol. And what's happening is that the liver is breaking down glycogen and making glucose 6-phosphate, where it's going to be released for peripheral utilization by the expression of glucose 6-phosphatase. So the muscle also breaks down glycogen, but it doesn't express this enzyme here. And so it keeps the glucose 6-phosphate for itself. So in the high insulin, low glucagon state, we're in the fed state, and any excess carbohydrates can be shunted off, where various tissues, including the liver, muscle, spleen, etc., can actually make glycogen. So glucose 6-phosphate is a central metabolite. What is it? It's an activated form of glucose. So you actually have to use an ATP molecule. You have to burn some energy in order to activate the sugar molecule. And then it has options. And we discussed this before. So today we'll look at glycogen synthase. This enzyme, as you might expect, is upregulated by insulin. And here we make a glycogen, which appears to be a polymer of glucose. We'll take a look at some more structural details of the glucose as well as the glycogen molecule in a moment. We talked about glycolysis. We know that glucose 6-phosphate carbons are committed to the glycolytic pathway through phosphofructokinase 1 enzyme. That is the rate limiting step. We also talked about the pentose phosphate shunt. This is a pathway that actually oxidizes glucose 6-phosphate to ribulose 5-phosphate. So you make CO2, but you also make reducing power in the form of NADPH, which we saw was used to actually synthesize fatty acids as well as cholesterol. So here's glycogen metabolism. So here you see a very shortened version of the glycogen polymer. And what I've done is I've started to label the carbons in the ring. You should be very familiar with this nomenclature. Here's the 1, the 4, and the 1. This is a 1-4 linkage. It's an alpha linkage because these bonds here in the glycosidic bond, so it's basically an ether, ROR. What they do is they're below the plane. So if you draw the plane like this, the oxygen lies below it. So below is alpha, beta is up above the plane. Here's the 1, 2, 3, 4, 5, and there's the 6. So glycogen synthase adds on glucose units, but notice the glucose itself is activated by UDP. So it's very common in biology to activate sugars through uracil derivatives. We'll talk more about this in a moment. We add on a glucose moiety, so we have 3 in the chain. And then phosphorylase is an enzyme that can degrade glycogen under conditions by which you'd want to mobilize stores for energy. This would be, for example, low insulin, high glucagon, high epinephrine. In the liver, it's glucagon is predominant, although epinephrine does work in the liver. Glucagon only works in the liver, and we've said that before. That's something you should definitely know. And so phosphorylase enzyme performs phosphorylases, as you see here. It doesn't use water as a nucleophile. It uses inorganic phosphate. And what happens, you end up with a glucose 1-phosphate at the end, plus the remaining glycogen entities here. So glycogen branches. So we've seen the 1-4 chain here. We also see 1-6 chains. And so branching serves an important function. So what you're going to get is a really giant branch chain molecule. So if we look at these linkages further, we discussed alpha 1,4. This is an acetal right here. these glycosidic linkages, carbon is in the form of an acetal. Over here, this is a hemiacetal. So let's take a look closer. So this is what an acetal is. You should definitely know this for the MCAT. This is a hemiacetal. And the distinction is that the hemiacetal is unstable. It's free reversible. And so we'll see in a moment that this side of the actual glucose entity here can open up, but these acetal linkages are unstable. It's not a reversible reaction. It's not an easy one to break either, for that matter. So what we have then through this is the concept of mutarotation. So let me take a look over here on the right, or excuse me, on the left. So here is the Fischer projection of glucose. By convention, the most oxidized carbon is at the top. And in red is the OH group, and this represents the D configuration. If the OH was on the left, it would be the L configuration. So we have some stereochemical principles to keep in mind here. Now, this OH is nucleophilic, and what it can do is attack this sp2 carbon here. This carbon's planar, and so the OH can attack from the top on the right, or it can attack from the bottom. And when it does that, it can push the hydroxyl down, or it can push the hydroxyl up. And what you see then is that these are basically isomers of each other. And so what happens then, because this is a hemiacetal linkage, it can open and close. So that ring is opening and closing and opening and closing. And every time it opens, there's another nucleophilic attack that could happen to it to close it. Well, it turns out that the vast majority of glucose molecules or monomers in solution are going to be in one ring structure or the other. So if you notice, the alpha here is 35%, the beta D is 65%. That's 100%. So essentially, this is a very, very vanishing small amount. It's always going to be in the ring structure, but it goes through an intermediate. So as it opens and closes, it's going to generate an equilibrium. And that equilibrium will be about 35% alpha D and 65% beta D, because the reason why it's beta D is more favorable is because the large OH is in the equatorial position, and that creates less steric hindrance than it does if you're in the alpha, if you're in the axial position, excuse me. So what we see then is the concept of mutarotation, where the glucose can open and close and open and close. Now, an important concept that's on the AAMC content outline is the thing called reducing sugars. So what we see is that if we look at glucose further, we actually see it's an aldehyde, and it's called an aldose. So carbohydrates with aldehydes are aldoses. And aldehydes, as you see up over here, can actually be oxidized to carboxylic acids. And so there's a solution known as Benedict's reaction or Benedict's reagent, and it oxidizes aldehydes to acids. And so what people can do is they can determine whether a sugar is a reducing sugar, because if it's a reducing sugar, it'll have a reducing end. What is the reducing end? Well, it's the carbon with the hemiacetal linkage that can open and close. And in the open state, you can oxidize it with this copper solution. So in the aldehyde form, it's going to be a blue copper. But when you oxidize it, you turn into this copper oxide, which is actually red. So the presence of red tells you that you had a reducing sugar. And you can use the Benedict's reaction outside of carbohydrates. That's its traditional role. So this carbon here is in an aldehyde. It's susceptible to nucleophilic attack. And when you do that, you generate the alpha or the beta. This carbon is said to be the anomeric carbon. So let's take a look at glycogen synthesis. We know it occurs in multiple tissues. We know that glucose 6-phosphate is derived from glucokinase or hexokinase. It comes into the cytoplasm, and then phosphoglucomutase can convert it into glucose 1-phosphate. And so this reaction is basically an isomerization reaction. So it's governed by mass action, or Le Chatelier's principle. So the more glucose 6-phosphate you have, you're just going to push the equilibrium towards having more glucose 1-phosphate. There's no energetic requirement in virtually all of the isomerization reactions. And then what we see is this glucose 1-phosphate uridyl transferase actually uses the energy of UTP to activate the sugar. So whenever you activate molecules, you typically get to hydrolyze some high energy bond. And so we release inorganic pyrophosphate. And then what happens is that the UDP glucose can be added on to a growing glycogen chain. And this reaction is regulated by insulin. So glycogen synthase is a heavily regulated molecule. Insulin, which we know is anabolic. Insulin wants to make macromolecules when you're in the fed state where there's a lot of sugar around. And so you're going to store that sugar in glycogen, whereas epinephrine and glucagon, which only works in the liver, is going to actually oppose the glycogen synthase reaction. So it's going to inhibit it. And down here, what you see is the actual formula where we have to put in some ATPs. And what you can see is that the synthesis of glycogen is actually endergonic. And over here, this UTP, adding on to UDP glucose here, this PPI is hydrolyzed into two inorganic molecules of phosphate, and you release even more free energy, further pushing the equilibrium towards products. The reaction's irreversible. So here we see the actual structure here. Here's UDP alpha D glucose. This is uracil. Here's the actual sugar, the ribose ring. Here's the two phosphates over here, and here's the glucose moiety. And so what you can see happening is that there's a protein called glycogenin. This actually nucleates the site of attachment of all the glucoses onto a glycogen. And what you get at the end is alpha D glucosyl glycogenin. Here's what it looks like. So here's glycogenin as a protein. And what you can do is you can covalently attach onto this protein a bunch of glucose entities, and you start branching them, and you get a really beautiful looking molecule here. Now, glycogen is actually stored in granules. You can actually see that this entity with this little arrow down here, this is called the glycosome. So people believe that the glycosome is actually an organelle, that glycogen particle with all the enzymes that make it and break it and stuff like that. What they do is they form an organelle. And you can actually see better, more advanced shots over here. The G stands for the glycosome, the glycogen granules here. And the liver in humans makes up about 8 to 10% is glycogen by mass. So you store a lot of glycogen. So we look at glycogen metabolism. Over here, we see glucose 6-phosphate to glucose 1-phosphate. We talked about reactions 1 and 2, and we actually make glycogen. This is all promoted by insulin. But what about when we want to break down glycogen? So what we do is we activate phosphorylase, which is reaction 3, and phosphorylase, as I'll show you, gets phosphorylated. It's active when it's phosphorylated, and then when it does, it takes off one of the glucose entities from glycogen to generate and release glucose 1-phosphate. There's also branching and debranching enzymes. They're not shown here, but so debranching enzymes are involved in degradation of glycogen, whereas branching enzymes are used when you synthesize glycogen. So let's take a look at the signaling pathways that regulate glycogen breakdown. So here is what we have is a typical 7-membrane G protein coupled receptor. This is a heterotrimeric G protein. We recognize that what happens is that these G proteins, when they're active, are going to exchange GDP with GTP. And in the GT bound state, they're going to be active. And so they are alive as long as the GTP is active. So once you hydrolyze GTP back to GDP, the signaling cascade is reduced. So this typical reaction would be in the liver because we have glucagon working on it here. And so what happens is that when you have the ligand bind to the receptor, what you do is you exchange the GTP and GDP, and you activate a molecule called adenylyl cyclase. And what that does, it makes cyclic AMP. This is cyclic AMP right down here. This was discovered by Sutherland. It's actually heat stable, excuse me. This is how Sutherland won the Nobel Prize. So what it basically does is you do an organic chemistry reaction here, and you form the cyclical version of it that you see here. We have this phosphoester bond formed right there. So GTP is involved in activating adenylyl cyclase, making cyclic AMP, which is a classical second messenger molecule. And what cyclic AMP does, it activates protein kinase A. It actually binds allosterically to regulatory subunits, releasing the free catalytic subunit. And what pKa does, it phosphorylates glycogen synthase. So this would inactivate glycogen synthase because under the conditions of high glucagon and epinephrine, we do not wish to activate glycogen synthase to make glycogen, but rather we like to mobilize glycogen by breaking it down. So pKa phosphorylates glycogen synthase, turning it off, phosphorylase kinase, turning it on. Phosphorylase kinase activates phosphorylase, and then phosphorylase performs phosphorylasis, not hydrolysis. And what you do is you actually release a molecule here of beta D glucose 1-phosphate, and then this can basically isomerize by phosphoglucomutase into beta D glucose 6-phosphate. And what happens is glucose 6-phosphatase, which is expressed in the liver, can actually dephosphorylate it, releasing a non-charged dephosphorylated glucose, which can now exit the cell into peripheral circulation. And then recipient tissues can use that glucose, like the muscle, and oxidize it to generate energy. So what we see is a signaling cascade where protein kinase A phosphorylates phosphorylase kinase, which phosphorylates phosphorylase, which performs phosphorylasis. It's like Peter Piper picked a pack of purple phosphorylated peppers. And with that in mind, let's prepare ourselves to do a couple challenging passages that will integrate our knowledge of glycogen synthesis and degradation with overall metabolism. So let's get going.